Hello. Today I'm going to tell you a bit about representing biological sequences. In particular, I'm going to try and explain how to represent DNA sequences in a numerical form such that we can use machine learning on them. So firstly, let me try and motivate why we need a numerical representation. Let's pretend we have this data that are about employees of some company, and they have names, genders, where they live, what their qualifications are, age, and salary. Okay. Now, this kind of data it's common, but it's not something we can directly put into a machine learning algorithm. To put it into a machine learning algorithm, we need to convert this kind of tabular data into a form like this, where each column, so-called a feature, is actually a number. Okay, and in fact, so each column also often we want them to be somehow normalized and to take care of units and things like that. Okay, now in this table as well, each column is a feature, each row is a particular instance, and this is what we often use as input to machine learning. And so given that input, we can do something like regression, where we take a real valued vector input and output a real valued scalar. Okay. Again, using that example data, on the x-axis here, we have age. On the y-axis, we have salary. And the data points that was in the previous table is plotted here as blue dots. And the machine learning task that's of interest is, after training, after fitting some line on these blue dots, we want to predict what is the salary of somebody at age 60, which is denoted by the red dashed line here. Okay, this example was taken from this textbook by Mark, Aldo, and myself. It's available freely online at mmlbook.com. Now, for biological sequences, in particular, here we have an example of a DNA sequence which has 4 bases, A, C, T, and G. This can be very, very long. For example, the human genome is 3 billion bases long. Okay, so now given a sequence like this, how do we convert that sequence into a numerical representation? And that's the topic that I'd like to walk you through today. Okay, we're not going to look at all possible ways to do it, but I'd like to think about the kinds of things that are potentially interesting to look at, and that has interesting research questions for a machine learning audience. Okay. Now, before we get into that, I'll have to give you a very, very short tour of biology. It's really, just a few slides. Then use some of that biological knowledge to manually design features, just like previously when we are converting postcodes in London to longitude and latitude, we use the knowledge that sequence of characters was postcodes. Okay. Then we move very quickly to this idea of, well, let's look at occurrences of things, and then we this is called counting and hashing, and we briefly talk about kernels and how to compare these types of things, and I'll end with only 2 slides about language models. Okay, so before you do anything else with these types of methods, you should first try doing something called hot encoding. Okay, so in this particular case, we have a 4-letter alphabet, which means we need to represent a 4-dimensional vector, because there's 4 possibilities, and you just, for example, associate A with 1, 0, 0, 0, and so on and so forth, like on this slide. Okay, now this is often quite good. You don't need to really know anything about the system. You can just represent it that way, and it usually works. Now, why this one-hot encoding and not something like representing A with 1 and C with 2, G with 3, and T with 4? An integer encoding, so-called, actually assumes that the distance between A and C is 1, and the distance between A and T is 4, right? And so the numerical encoding actually captures the distance also of these categorical objects. Okay? That's one thing, and you may or may not want that. And in terms of DNA sequences, a priori, you don't have that information. There's also an interesting order going on, which is that if you represent A as 1 and C as 2 and G as 3, then you're making an assumption that G is bigger than A in some sense. Okay? And remember, when you plug algorithms, when you plug representations into a machine learning algorithm, you are actually encoding all the knowledge you have, and sometimes the unconscious biases that you already have. And so that's why integer encoding is not a good thing. Okay? Now, the other reason why one-hot encoding is quite popular is that once you encode things in ones and zeros, the data type is binary, which means they can be stored in 1 bit. And in fact, by using compression techniques, you can actually store quite complex, quite big data sets quite efficiently by doing things like run length encoding, just very, very simple things. Okay? It's worth also mentioning that you can encode labels this way as well. Okay, because this is also standard. The most pieces of software have an implementation of this, so I highly recommend just using a standard one instead of implementing one yourself. Okay, here are some examples from the Python ecosystem. Okay, so let's briefly talk about biology and use this very brief knowledge to construct features, construct numerical representations of the system that we care about. Okay? So this is a cartoon of DNA. DNA is actually a physical object in you. So in each of us, there's millions and millions of cells. In the middle of each cell, we're something called a eukaryotic organism. So in the middle of cells, we have something called a nucleus. Inside the nucleus is something called a chromosome. Okay. Now, the chromosomes are where it's just basically sequences of DNA, right? Now, what's important to remember is that the chromosomes, which are inside the nucleus, which is a super, super tiny object, you need a microscope to look at them. The length of your DNA in each of your cells is roughly 2 meters if you stretch it out. Okay, it's all packed up in this really intricate way, and this has quite interesting implications in biology, which we're only today just scratching the surface of. Okay, but DNA lives in a chromosome, lives in chromosomes, and which is in the middle of every single cell in your body. Okay? So there's an idea called the Central Dogma, which, you know, like many ideas in biology, as we progress in our knowledge, changes a little bit all the time. But the general idea is you have something called DNA, which is what we looked about, this physical object. It's written left to right. The left side is called 5', and the right side is called 3'. And the letters are A, C, T, and G. Okay. DNA is transcribed. There's a process involving a ribosome. It's transcribed into pre-mRNA. Okay, we're just going to call this for computer science purposes, we're going to call everything mRNA. There's a lot of different versions of it, but where the letter T becomes a U, Okay, but it's still a four-letter alphabet. So by and large, from a data analysis point of view, we're not worried too much about it. Okay, and then the mRNA is spliced, so certain parts of it are cut out. This process of cutting stuff out is called splicing, and it's something called introns that are cut out, and so you end up with this shorter sequence or mRNA, and which is then translated into proteins. Okay, proteins are three letters of mRNA get converted into 1 letter of a protein, and then an amino acid. Proteins have a 20-letter alphabet. Okay. Now, this is clearly a very cartoon picture. There are lots and lots of books written about this, and as I mentioned earlier, as we discover more about biology, a lot of this knowledge is being written as we speak. Okay. So now let's zoom in a bit on exactly one problem, which is to detect something called an acceptor splice site. Okay. It turns out that almost all acceptor splice sites have this tumor. It's called this two nucleotides, which are conserved. Okay, our acceptor splice site. So on the left of this picture here are introns, and on the right is where the exon is. And if you take all these objects all over the genome and stack them up on top of each other, you can see that at the boundary there's this conserved 2-letter alphabet AG. Okay. Now, what's interesting is that even though every splice site is centered at AG, not all locations which are AG are splice sites. So basically, 1 in 16, because there's 2 letters each with 4 possibilities, so there's 16 possible tumors involved. So 1 in 16 possibilities on the genome, assuming it's uniformly distributed, are potential splice sites, but only a very few of them, very rare ones, are actually true splice sites. Okay? So now, if you want to cast this as a machine learning problem, how do you detect which of these sequences are actually splice sites? Okay? So we can use knowledge, so biological knowledge. Okay, so we've known for a while that exons, the part that encode for proteins, have higher GC content than introns. Okay, we also know that the exons encode proteins, and so we can look for these codons. Remember, codons are these 3 letters of DNA that get translated into a peptide. Okay. And in fact, because the translation is actually executed by a physical object, this physical object recognizes certain sequence patterns near the splice site to be able to recruit this splicing machinery. Okay, and we can watch this brief video there. Okay, now, collecting all those little pieces of information, right? We can try and manually construct, these kinds of data. Okay. So, for example, here each row here is a particular sequence. Row number 1, with example 1, you look at the GC content before the AG letter. Okay, you just take any sequence, you put the AG in the middle, you calculate the numbers of G's and C's versus the number of A's and T's. So there's only 4 possibilities. So you can look at in that first row and the second column, you get 0.6%, 60% of that sequence is GC, and after the AG, it's 0.7. Okay. You can also look for motifs. Okay, so basically you can look to see whether at the AG location, I think the AG is at the end here, then did that sequence AG, AG, AA appear just before the AG, okay? And then you can just have a binary indicator, did that sequence appear or not, okay? Or you can look for some other sequence, and if you stare at the example that we have up there, just to the left of the AG, there's a whole bunch of T's, and you can say, oh, okay, that's kind of interesting. So let's try and detect that feature, that motif that existed. So that's the column TTAG, TTTAG, right? And along with that, we have biological experiments that tell us whether this was truly a splice site or not, and we can use this as a label. Okay? Now, it's worth again reminding you that the number of AGs that you have is 1/16th of the genome. Only a very, very few number of them are actually true splice sites. Okay? So the proportion of positives is actually very small. It's a very, very imbalanced problem. Okay? But we can use our domain knowledge. I just, from these few minutes of talking about biology, we can construct a matrix like this, and then we can take this matrix and plonk it into any machine learning algorithm that we like. Okay. So while we were talking about this, we were looking at these objects, which are essentially subsequences of a sequence. Okay. Now, there's this really interesting concept, which is about the occurrence of subsequences, and this is what I'd like to spend a little while talking about now. Okay, so using this idea of what is the content of something, you can think about, well, why not longer letters? So why not more letters? Sorry. Okay. So instead of using just one letter, you can use the number of substrings as a feature. Okay. So here's a worked out example of this object for tumors. Okay. Sorry, I should have mentioned that the number of substrings is called k-mers in computational biology. It's very similar to n-grams in natural language processing. Okay. So let's look at this sequence. So GA, TTA, CA, A. Okay, you take this sequence and you take a window of length 2 and you slide it along the sequence, and for each occurrence of this tumor, then you add it to a table. Okay, so for example, here we've created a table. This is the set of all possible tumors, AC, AG, AT, and so on and so forth. There's 16 of them. As we slide it along, the first tumor is a GA, so we add 1 to the GA column, and then the second one is AT, we add 1 to the AT column, and we slide it along, so on and so forth. Okay, and as you see, there's a whole bunch of A's at the end, and that means there are 3 tumors which are of AA. Okay, so this is a very simple idea. Okay, and the tricky part is that there's really a lot of these substrings. Okay, so with tumors we've got 16, but if we're for many of the problems that we are interested in, it turns out that we need substrings of quite long lengths. Okay, so 21 seems to be a very popular number. Okay, and so there's really quite a lot of them. Okay, before we try and address this question of what do we do with this many substrings, I'd like to do a little bit of a segue to discuss somehow a philosophical issue. Okay, the philosophical question is, in genomic systems, it's like coordinates are kind of really tricky. Okay, so we tend to use coordinates really commonly in machine learning. Okay, for example, in spatial-temporal systems, if we're doing mapping, we use longitude and latitude for locations of Earth. So if we have a particular object, we can locate it by the coordinate system that places it on the surface of the Earth. Okay, of course, if any of you who works in GIS will know that this is not so obvious how to do, but because the Earth is not round, but by and large, we have that coordinate system. If you're looking at the sky, if you're an astronomer, you use a very similar kind of coordinate system, but you call it right ascension and declination. If you're working on object recognition, you tend to use the pixel locations, but then these pixel locations also map to locations in the real world, and that mapping is not always obvious, and figuring out how to do that mapping is an open problem, right? In mathematics, right, we use coordinates all the time. Okay, so if you have a vector space, how do you represent the coordinates of a vector space is actually an interesting question. So most of the time we just ignore it. We use the standard basis, it's called. But by and large, behind that, you actually have to construct what this coordinate system is. Okay, so and the way you construct it is by thinking about what's the vector space, and basically you think of a set of vectors, here denoted by curly A, which allows you to reach any point in your whole vector space by a linear combination. Okay, and this set is called a generating set. Okay, and when you choose a set x1 to xk that is as small as possible, then we have something called a basis. Okay? It just means that it allows us to refer to any point in the vector space in terms of this set x1 to xk. Okay. Most of the time, we tend to use orthogonal, or orthonormal basis in representation, but in general, you just need some set to talk about it. Okay? Now, the interesting thing in biology is that you don't actually have a very natural way to talk about coordinates. Okay. And in fact, one of the hard tasks in biology, when you have a new measurement of the world, you have a new sequence that you care about, you actually have to do something called sequence alignment, which I'll talk about at some other point, to figure out where this sequence came from. Okay? And essentially, you kind of have to do some sort of pattern matching to find the right place to put this object. So there's no real good way to index it. And this is the source of a lot of difficulty in biology. Okay. So it turns out that there's a parallel path of research, which is, why k-mers are important and interesting. So there's a lot of progress in that, because it turns out that sequence alignment is an expensive process, and so people want to do alignment-free sequence analysis. I'm not going to talk about that at all today, but just to point you to that literature if you're interested. Okay? So now back to this question of we've got a lot of k-mers. Okay, so we're interested in substrings of DNA, and we want to count the occurrence of the substrings. And it turns out there's a lot of these substrings. Okay, so if we take a k-mer, we measure something in the world. This is an output of some machine. It's called next generation sequencing right now. It's already more than 10 years old, but we still call it next generation sequencing. We get this file from this computer that reads the DNA, and using that, we count, we call it the k-mer count, we count the number of times the subsequence appears in these reads. Okay? Once we have these counts, we have this vector of numbers, and we can compute similarities and inner products and kernels and things like that. I'll come back to the kernel a little point later today, but for now, let's focus on the counting. So here's our favorite sequence, GATTACAAAA, and we want to count the 2-mers, right? So we slide a window across this sequence, and we add 1 to the relevant column when it's needed. There are 16 possible index values here, okay? And we've essentially invented a coordinate system for DNA by saying, you know, that's the way we want to do it, right? That's the way we want to order our substrings, okay? And that's one observation. There's 16, and as the k-mers get longer, that sequence grows really quickly, okay? The other observation is that there's actually many entries in this array which are 0. Okay, and it turns out that as you make the k longer, as the k-mer substring that you're looking at becomes longer, then the number of zeros actually become more common. So this data is very, very sparse. Okay. Okay. Now, why do we want to do this representation? Once we have this representation, we might want to compare 2 sequences. Okay, one thing about sequences is that they could be of different lengths. Okay, so we have one file that comes from a DNA sequencer. We call it a read. It's our favorite sequence, and we have another file which is almost identical to it, except it's got 2 C's on the end. Okay, now the 2 representations, so the 2 k-mer count arrays of these 2 files are shown just below, and you can see that those 2 C's on the end added an AC and a CC. Okay? So given these 2 sequences, we can compute a similarity by converting them into these arrays of 2 counts, and then we can just use the standard inner product between these 2 vectors. Okay. Now, there's an assumption here that we indexed the counts in the same way in those 2 arrays. Okay, so in other words, we use the same way to order our substrings. Okay, that's one important thing, and this goes back to that philosophical discussion about coordinate systems. Okay. The other thing is that if we are interested in computing an inner product, basically we're just comparing, going back one slide, if you're just comparing the letters, the substrings, right? You're comparing each column of this array, and you only need to compare when both of them are non-zero, all right? Because as soon as one of them is 0, then that entry, the product of those 2 things are 0, right? Okay, so we only need to consider the indices where both are non-zero. Okay, and it turns out that you can exploit this to count things a lot quicker. Okay, we'll come back to this a little bit later. Okay, so we have a sequence, and we want to do this representation like we have here in this picture here. I've explicitly written down the index. Okay, so you can think of this as the number, the location of the position. I've started my counting at 1, and it goes up to 16. Okay. Now, if I wanted to fit this array in memory, I tend to need a lot of memory. Okay, as I said earlier, seems like in a lot of applications, we want to use quite long subsequences, say k is 21, and the length, the number of possible substrings is 4 to the 21, which is roughly a terabyte large, and this is not quite something we can quite do yet. Okay, so for the next few minutes, I'm going to tell you about this way to convert, to count substrings, but still keep that array in memory. Okay? There's all sorts of interesting tricks coming from computational science about how to do this really efficiently, and it's coming from the literature of streaming data and online learning and things like that. Okay. Okay. So I've just copied that count in that first picture there, and the key idea of hashing is to just use a smaller array and hope that things work out. Okay. Now, like I've chosen this smaller array in a very special way. Okay. Now, what I've done is my original array was length 4 to the 2, 4 squared. It was length 16. And I've just chosen a smaller array of length 7. Okay. And what I've essentially done here is just wrapped around the array the counts that are afterwards below the first one. Okay, so the first 7 indices have been just copied, and then index number 8 to 14 gets copied just below, and 15 and 16 gets copied just below that. Okay. Now, this trick, it's called modular arithmetic. Okay. Modular arithmetic is something called, you can use modular arithmetic as a hash function. Okay. And then your counts, if you counted it that way, you can get this smaller array, and you get the counts as follows. Okay. One thing that has happened by doing this wraparound is that we've now lost our ability to know which index, so which location in the array means which substring. Okay. You can still work it back if you keep track of this in modular arithmetic, but by and large, we don't really care about this, and we just forget about that. Okay. Now, the interesting thing is once you've done this, you can still do inner product. You've got an array. If you've noticed, there's some difference in this, and the question is this a good representation? Okay. So for those of you who've been paying attention, let's go back to the previous slide. For some items, like the item number 13, okay, so TA, that gets wrapped around, and when you do modular arithmetic, it ends up in 6, and that count is captured. Okay, so there's kind of no loss of information in some sense, right? But for the AC, it's in the index number 2, it's clashed with index number 9 and index number 16. Okay, and this clashing in the literature is called a collision, and this is generally a bad thing. And when people design hash functions, they want to design it such that collisions don't occur. We are interested in computing the inner product, and so if we don't have collisions, things are accurate. Okay, and there is great hope that you can design something where collisions don't occur, because remember that the number of non-zero entries is actually quite sparse. So most of this array is very empty. It's very hard to show with k-mer of length 2, but if you have higher k-mer lengths, it turns out that it's really mostly very empty. So there's all these zeros that you can reuse. Okay, we did this work we computed this k-mer based counter, and we computed this in a product. And it turns out that we can look at the entropy of the particular entry, and this improves the performance a little bit. We call it the k-mer weighted in a product, and this runs really quite nicely. Okay, and we applied it to some quite nice applications in rice, Chlamydomonas, which is actually pond scum, and looks at relatedness of metabiomes. Okay? Just to give you an example of the kind of result we got in that paper by doing this kind of hashing count of k-mers. Okay? On the left is our result. What we did was we took the read file and we counted k-mers in this hashed way. And because we have a vector, we can do standard machine learning, and we did PCA and plotted here the data points. Each dot here is a particular organism. The two axes are the first and second principal components, and we've plotted them there. And the colors are basically the location of the where they got the sample of the pond scum. Okay. Now on the right, we have a previous implementation where they took these sequences. They had to do some very complicated analysis. So they had to detect something called SNPs, which are singular nucleotide polymorphisms. You look at these variants, and then there's this very complicated pipeline to identify the right variants. And after that long pipeline, they did PCA. Okay. Now, if you look at these two plots, they're very, very similar. The one on the left is purely just counting occurrences of substrings. The one on the right uses significantly more information to try and recover the variation between the different organisms that are measured here. Okay, so that was quite nice. The question remains of how do we reduce these collisions? Okay, it turns out that modular arithmetic is really terrible. Actually, it has really, really many collisions. And in that work that we presented, we used something called murmur hash. The reason it was chosen is because it's really, really fast. It's really only something like 10 assembly operations. Okay, so it's really down in the chip. Okay, so if you have a good hash function, and there's a lot of very interesting work around hash functions, you can do this. One thing worth mentioning is if you look into the literature of hash functions, is that a lot of hash functions are designed to be something called cryptographic hashes, which means that the hash functions are secure in some sense and provably secure. In this particular case, we're not so interested in the security. We're more interested in how fast you can hash the subsequence. Okay? So there's that. So there's also some interesting research like that. And there's another direction of research, which is if you have a particular hash function, it will result in collisions. And in some sense, collisions are not avoidable, right? You have this gigantic long array and you're reducing it to a smaller array. Collisions are somehow not avoidable. But you could exploit the fact that different hash functions will have different collision patterns. Okay, so you could use more than one hash function. Okay, and this is a very, very booming area of research. Highly recommended for those who are interested in kind of large scale sequence analysis. You can look at hashing. This is idea called a Bloom filter. You can do hashing such that sequences that are very similar to each other get hashed to nearby locations on the array. Okay? So this is called locality sensitive hashing. And there are two, kind of famous objects that were invented almost 20 years ago now, right? The hyperlog and the count-min sketch, which allows us to do this counting really efficiently. In the last 2 years, there's been this real interest in something called the race sketch, which combines the idea of locality sensitive hashing and multiple hashes at the same time. For those who are interested, I highly recommend this direction of research. So we can now do this hashing, and at the beginning of this part about counting substrings, I did mention something about kernels, and I'm going to change gears now and speak a little bit about what it means to take inner products. Okay. So what is an inner product? Here's the mathematical definition. You have a bilinear mapping. So this is a function with two arguments denoted by dots here. This mapping between x and y is something, when you say it's bilinear, okay, it means that it's linear in each of the arguments by themselves. So for example, which means you can pull out scalars and you can add things together. Okay? Like for the first argument, it's shown here. Okay? If you have a bilinear mapping where you can flip the order of x and y, it's called symmetric, and that it's positive definite, then it is an inner product. Okay, so this is the mathematical definition, and most of the things we care about tend to be in this form. Now, if we if x and y are d-dimensional vectors, if x is an array of numbers, right, and you do the standard inner product, the standard dot product, then you have this inner product. But by and large, they're more general objects. Okay. Now, what is a kernel? Okay, a kernel is when you take an inner product between some version of the object itself. Okay, so I've got two objects, x and y. They may not be in a vector of numbers. They may not be represented as a vector of numbers. Okay, so on the left of the equation, I've got a kernel between two objects, x and y. They may not be. They may be DNA sequences, for example, right? I take a mapping, and this in the kernel literature, it's called phi usually. It's called the feature mapping. So I take the object I'm interested in, say the DNA sequence X. I map it into some sort of vector, like we did with k-mer counting, and then I take the inner product between the two vectors. Okay. Now the key insight of kernel methods is first that you can compute, similarities, so kernels between objects that are actually not even vectors, but somehow under the hood, you're always really computing this vectorial representation. You may not explicitly compute it like we did, or you might be doing some sort of funky trick like hashing, but under the hood, somehow you always have this vectorial representation. Okay? But it turns out that computationally, you can compute the object on the left, so which is the inner product of the mapped objects. Okay? Sometimes you can compute them faster than you can actually do it by doing it on the right side. So when I say faster, I mean the number of computational operations for computing the similarity is smaller than actually representing the objects as vectors and then computing that inner product. Okay, so, and a bit of intuition why this is possible at all is, for example, in our k-mer counting application, most of the entries are 0. Okay, so when we do compute the inner product, as soon as one of the entries are 0, we never need to compute that contribution to our kernel, right? And so when we index this object, we only need to keep track of the things that are non-zero, and this can be significantly faster. So imagine you've got a super long vector, 1 terabyte in size. Everything is 0 except the first entry. So to generate that vector would take a long time, but to allocate the memory for that vector would take a long time. But if you knew that only the first entry was non-zero, when you computed the kernel, you only needed to just keep track of that one single number, right? So that's kernels. Okay. Now the k-mer counting approach turns out to be exactly something that is called the spectrum kernel. Okay, so what is the spectrum kernel? You count a particular k-mer in a sequence as a feature. You represent that feature map, which we discussed before already. And the spectrum kernel is literally the inner product of these two feature vectors. Okay? And this was invented in the early 2000s, 2001, I think, and which means, and this, the realization was actually you only need to look at the substrings that occur in both sequences to be able to compute this inner product. Okay? So that's the spectrum kernel. And so in some sense, all this new work on vectorial representations are somehow recovering a lot of the early results on the early 2000s. Okay? And so when you look at the spectrum kernel and you can look at properties of positive definiteness, you realize that the weighted sum of kernels is a kernel, and you can look at mixed spectrum, so which means that you can look at substrings of different lengths and you can just add them up. Okay? This is a figure from the tutorial paper that it turns out that you can combine these features and improve your classifier. Okay. So what is being shown on the plot here is something called an ROC curve, the receiver operating characteristic curve. Okay, so if you look at the GC features, just like we had right at the beginning in an example, the GC content just before the splice site and the GC content just after the splice site, and you try and classify whether the object is a splice site or not, you get the ROC curve, like a black line. If you use a spectrum kernel, you get a slightly better result. And if you look at substrings of length 3, you get an even better result. Okay. One thing that we haven't touched upon, and one thing that spectrums kind of forget, okay, is that the actual location of the substring is important. Okay, so somehow I said earlier that genomes don't really have coordinates, but on the other hand, they have relative coordinates. Okay, so again here we have this AG consensus sequence just before the splice site, just before the acceptor splice site, and if we look at the occurrence of other letters of all the splice sites. So what you do is you download all the splice sites that have been empirically verified, and you align it around the AG. So the splice site is position 0 here, and you look at the proportion of letters. You know, how many of the letters at position -5 is T. Okay. It turns out that a lot of them are in RT at position -5. Okay, so 5 letters before the splice site, it's almost always a T. Okay, the y-axis here is the number of bits for that in terms of entropy. Okay, so we really kind of want to capture that position. Okay, it's not an absolute coordinate, but it's a relative coordinate, and this is kind of interesting, right? Okay. And to do this, you can do something called a weighted degree kernel. Okay, and the way you do this is you take two sequences, you identify the largest matching blocks for their sequence. Okay, and for each matching block, you add it to the kernel. The thing is that you weigh the blocks differently according to their length. Longer blocks are rarer, so you want to give them more weight. Okay, and you can think of this as the spectrum kernel. So just the k-mer counts, but then for every single position, so you just copy the spectrum kernel for every possible position and weighted by the length of that spectrum. Okay, and in fact, you can even allow that these matchings are offset from each other. Okay, if you're interested in this kind of thing, there's this tutorial paper we wrote in 2008, which talks through these kernels. Okay. Now, changing gears a bit, one thing about inner products is very often people get confused about distances and kernels or inner products, right? Intuitively, they are somehow in opposite directions. Distances are bigger numbers mean things are more different. In kernels, in inner products, in similarities, bigger numbers mean that things are more similar, right? So intuitively, they're somehow in opposite directions. Okay, you can convert. So if you have a kernel, you can always get a distance. Okay, but not the other way around. Okay, so they're not exactly opposites of each other. Most people think they are, but they're kind of just subtly different. Okay, so if you have a kernel, and if you have in a product, you can compute, you can define something called a norm, and the norm can be expressed purely in terms of the kernel, and then you get this expression as below. Notice there's a typo there. So the middle term, it's kyy, not xy. Right. The other way around is not always possible, but if you do have a distance, then you need to get a kernel if you want to get an inner product, something like you want to use a support vector machine. Then what I recommend is basically take the negatives of the squared distance and scale it appropriately, and that's a valid kernel. Okay, so if you're interested in that other direction, it requires a little bit more machinery, but by and large, you can always just try the exponent, and that should usually work. Okay, I'm almost at the end here. I'm just going to quickly mention something that might already be obvious to most of you. Okay, so k-mers in computational biology are very similar in idea to n-grams in natural language processing. Okay, so in the sequences we've looked before, we look at these 2-mers and we just count the occurrence. In natural language processing, we try to count words. In fact, you can also count something called charactergrams, right? So you can just look at windows of particular subsequences and just count them. Okay? These charactergrams can be used, for example, to represent new words or names or things like that, which are not real English words, right? So there's this obvious similarity. Now, given the huge progress we've had in the large language model literature in recent years, there's this really interesting open question of, can we use these language models on DNA or proteins? Okay, and just in the last year, there's just been lots and lots of papers about this, and here's just a selection that I found by looking online. Okay? So there is somehow a similarity between the way we represent DNA and natural language. We can embed, we can represent these biological sequences into a feature space. It's very similar in the way that we are representing natural language. Now, there's a caveat, though. When we are representing biological sequences, we don't actually have a very intuitive way to interpret whether a particular sequence is meaningful or not. Okay, and so that's why it's an open research question. So for those who are interested, I think it's a great place to work. Okay, let me summarize what we talked about today. We looked at representing biological sequences. Before you try anything else, try one hot encoding. DNA and RNA sequences are 4-letter alphabet. Protein sequences, which we didn't talk about at all, are 20-letter alphabet. You can use your knowledge of biology to design features by hand. Some of these features are essentially counts of substrings, and the counting of substrings allows us to avoid this question of the coordinate system of the genome, and so this is kind of really nice. But if you're going to count substrings of a longer substring, so say 21, you end up needing an index that's too big, and to avoid that, you do hashing. And there's very, very nice literature about hashing. Turns out that counting k-mers is very, very closely related to something that was very popular 20 years ago, which are these kernels on spectrums of DNA. Okay, and maybe today we can use what we know about large language models to attack these problems in biology. So I look forward to lots of interesting results from all of you. Thank you for your attention.